to have Scott McNeil, who will go on more detailed aspects of the NBCDs, nanoparticles, from his experience in his lab. Scott.
Okay, thank you, Stefan. Very good discussions indeed. We don't often get into the nuances and details of the pharmacology, especially when you're dealing with nanoparticles. So believe it or not, the example you gave is a simple one compared to a prodrug that has its own release kinetics, that has its own degradation. So, okay, so if you're familiar with NCL, I'm not going to discuss my laboratory for a change. I've done that 5 years in a row. If you need more information on what we do and how we characterize, just go to ncl.cancer.gov. So I've been asked to talk about NBCDs and more specifically critical quality attributes, CQAs of NBCDs. So the first bullet you see there are for the innovator, where they've developed a new concept and they're trying to pre-guess, if you will, what the regulators are going to want to see in that IND submission. So the onus is on them then to figure out their CQAs. The second bullet is kind of like what we just discussed, is for the follow-ons. Are we sure that we know which CQAs to look at, because we can definitely generate a whole lot of data, but is that absolutely germane to the mechanism of action, especially if the mechanism of action is not clearly defined, and it can be a very complex drug, such as the case with Copaxone. Okay, you saw this in one form or another throughout the morning's talk. So a CQA is some attribute that influences a biological effect. That can be safety or efficacy. It must be clearly defined, although clearly defined, as you see in the definition there, especially in nanomedicine, we cannot often clearly define our nanomedicine. So for a small molecule, we can say it's exactly 524.23 molecular weight. We know exactly what it's supposed to look like on NMR. That is absolutely not the case for most nanomedicines. You'll also see that we have the ability to define a range. So a range for an imaging agent that needs to be excreted through the kidney. Part of their CQA is going to be, well, it's less than 9 nanometers. But what does that mean? Does it mean 8.95? Does it mean 7.65? And so there's a series of experiments that can be done to define that range, as opposed to a very discrete number, such as is the case with small molecules. I'll also say what I've heard at least twice this morning is that every particle is unique, every concept is unique, and it needs to be treated as such. So it's, unfortunately, we don't, with very few exceptions, we don't have just simply a manual that you can take off the shelf and say, aha, here's my CQAs because I have a liposome with a drug in it. No, chances are that they're going to have to be defined. As I just said, they're unique. So you can take prior drug knowledge for things like liposomes, but if you have a polymer that's a prodrug, it is very difficult to know a priori what those CQAs are going to be. And so then we also verify with biological testing. It's a cyclic process where we get a result, we feed that back into our evaluation, and also to look at the physical chemical properties that influence that result. So for example, if I have a PEG density on the surface that influences phagocytosis, I can now define that through a physical chemical property, a PCC property, and define that for my process development as we manufacture the concepts. Okay, so much for small molecules. Now it gets very interesting with NBCDs. So by definition, the C of NBCDs means that it's complex. It means that often we cannot fully define it through physical chemical means. because the excipients, or what used to be called excipients, can actually contribute to the API, to the overall effect. They can influence things like drug release properties. They can influence biodistribution. And so where previously the API was that small molecule and there was just kind of some stuff in the tube, in the vial with it, now the whole package is often defined, or has to be defined, in NBCDs. Okay, so we used to have two buckets, one for the small molecules and generics, and then for the biologics and biosimilars. But in the case of NBCDs and nanomedicines, it can lie somewhere in between. Okay, so based on the 400 particles that we've seen come through the NCL, here's a quick list on some of the things to look for as you're defining your CQAs. So one is the polydispersity. What are the upper bounds and lower bounds, and how do they affect drug distribution and biodistribution? So the example I give you here, by DLS, that drug or that formulation looks like the red chart there. There's two populations. But which one has the drug in it? Because those will have different biodistributions. And so we have a number of techniques to look at that now. In fact, if you could, at the very exact same time now, Jeff Clogston from NCL is speaking in the other session on exactly this point. Okay, on surface coating, I've given you a number of examples over the past, and you probably recognize this one from last year, where the surface coating is probably the most critical factor that we go after in NCL, because it contributes so much to things like distribution, phagocytosis. It can contribute to toxicity and to overall efficacy. Here's an example that I gave last year where we received two batches of material. The first one had granulomas. The first one, this one's, I think, swapped. This example swapped. In the first batch, we saw quite a bit of granulomas, and we found out that was due to the PEG density on the surface of the gold particle, because the second batch had hardly any. So they were able to use this attribute, meaning the density of that PEG on the surface, as a CQA. This one was just very elegantly discussed in the previous talk. What we see, and just to confuse you more as far as drug release profiles, is sometimes we see that the free, well, let me back up. Okay, so we have encapsulated, we have free, and we have protein bound. Okay, but in the case of something like Abraxane, where it's already protein bound and it came in two different sizes, because it's introduced at about 110, and as soon as it hits the bloodstream, it goes down to 15. I'm off by a little bit, I think. There was a case where a follow-on used Abraxane as the reference listed drug, but the follow-on was a polymer. So imagine trying to compare apples and oranges in that case. Nevertheless, they got through phase 3 and did fairly well. So there are we have seen some very interesting kinetics where there's a release of the drug from the nanoparticle, but there's also a backward equilibrium back into the particle. It's lovely. Never a boring day at NCL. Also, we pay attention to impurities. We found that some of the impurities, the surfactants specifically in nanomedicine, can be very toxic. What you're seeing there is what we found, that when we initially tested these gold particles, as most of you know, gold is very inert. It's benign. We should not see any toxicity whatsoever, and it had very severe toxicity under our in vitro conditions. It turns out that the retentate, meaning what was left over after we spun the gold down, was the toxicant. So we encourage our sponsors, our clients, to look at all of the components, and especially impurities, if there is a surfactant that is needed to make the nanomaterial and to keep it from aggregating. Also, we go after stability. It is this bed is too hard, this bed is too soft, this bed is just right scenario, where if the concept is too stable, meaning that it's covalently linked, it'll be very, very safe, but it'll be very ineffective. Whereas if it's unstable, we get a burst of bolus release, and that in itself can have off-target toxicity. So the just right is being able to tune that, and then relevant to this discussion, defining the CQAs that gets it just right. How many springs have to be in that bed? How much down quilts have to be there? Okay, and then again, each particle is unique, each concept is unique, and even each batch is unique, as I showed you just a minute ago. So in the CQAs, we run it several times, and we don't make strong conclusions just based on one batch. I have 2 case studies. This is very similar to what I just showed you with the granulomas. The point of this is that we use multiple orthogonal techniques to go after an answer. This is an in vitro phagocytosis assay, and as you can see, both of those compounds, meaning even the pegylated, was phagocytosed to the same extent, or not phagocytosed to the same extent as the control. If you just took this alone, you would say that they're not phagocytized. But oh, wait a minute, let's look at EM first before we make that conclusion. And you can see by EM, the black marks there in the top are the particles that have been phagocytosed, and at this specific time point, the ones that pegylated were stealthy. They avoided phagocytosis. So again, use orthogonal techniques. Here's an example of knowing what the true measurand is as you do analysis. These are commercially available gold particles, and as you can see there by EM and AFM and DLS, they look all the same, but those are in pristine environment in something like PBS. But look what happens when you stick it in plasma. You get opsonization of proteins onto the surface of the gold, but you cannot pick those up by EM or AFM. You see that the size didn't change with those two, but the size with DLS, which measures hydrodynamic radius, more than doubled. Okay, just summing up, things to look for are really, many of them are common sense. Look for endotoxin contamination. You may think that is common sense, but 1/3 of compounds that are submitted to NCL still, to this day, 14 years in existence, are disqualified due to endotoxin contamination. 30%, 1 out of 3. We can also do quite a bit of in vitro, in vivo correlations, especially with immunotox, things like cytokine release, hemolysis. And I'm just going to go through this very quickly, where we also attempt to identify what the toxicant is in a specific formulation. In this case, it turned out to be the nickel, the cofactor, I guess, that you use to load it. And we were able to identify that as the toxicant, again, going back to the point that we monitor all of the excipients. I'm getting screamed at here by this monitor. So in conclusion, we know that physicochemical properties greatly influence biocompatibility. Go hard after those, but as you go hard after those, remember that because they're NBCDs, they're very hard to define, and don't rely exclusively on PCC, as was said earlier. Make sure that you use proper controls when you do this, because often nanoparticles can interfere and inhibit or enhance the assays in addition to the normal positive and negative controls, and use relevant buffers such as plasma when available. Okay, here's my contact info. Each and every year, every time I get up here, I say one thing, and that is you're doing great work. I want to encourage you, having seen 400 different concepts, you're in a terrific field. 14 of those are now in clinical trials, so keep it up. I always say that in the case of my laboratory, under the National Cancer Institute, cancer is no respecter of boundaries, borders, tariffs, or tweets. Okay, there's my contact number. Thank you.
Thanks,